Hello, everyone. We're going to switch to English again. My Dutch is worse than Lucy's, so sorry for that. Bear with me. Today, I'm going to talk about lipid residue analysis on archaeological pottery. Of course, there is a lot to cover on this part, but I will try to be very brief and to the point, and I want you to have an idea of what's and why's on this topic. So this is an outline of what I'm going to talk about today. I'm going to talk about what is lipid residue analysis on archaeological pottery, why do we use it, and what can be identified through this method. And after that, I will give a very brief information on sample selection, sampling, sample storing. I think that is very important also for the depots. Lab analysis, data processing, interpretation, and the limitations of the method that I used. So, organic lipid residue analysis actually means that we're working on the food residues that got into the pottery during processing the food, cooking or storing, that are not invisible to the naked eye. The visible parts that Lucy explained for you, the food residues, food crust, and lipid residue analysis, we go a bit more deeper than that, and we analyze the ones that are not visible to our naked eye. Otherwise, we would not know that are there. Organic residue survives in three main ways in archaeological pottery, or in archaeological context in general. One is the soil that's in situ, that comes from the pot. You can still analyze that, and it can give you some information through lipid residue analysis, but that is not the most common way of doing it. The second part is the carbonized surface residues, that is food crust that Lucy explained in detail, that are visible on the interior and exterior of the pottery, and that will give you an information on what is cooked on the last part of this pottery use. That could be related to one or two meals that is cooked in the pottery. And the third one is the observed residue, that is the fat that got into the pottery fabric from the food that is processed in the pottery. So this is the clay cups, cooking pots. Sometimes it is also on the glazed pottery. It is also a bit tricky, but observed residue ones are the most common ones because they preserve for a very long time in the pottery. So this is the one that we are analyzing most of the time, and this gives us actually unlike the food crust material, this gives us an information on the whole lifespan use of pottery. We cannot identify one specific meal that is cooked in the pottery. We cannot really tell you if they cooked fish and the meat and the cereal separately, but we can tell you what has been cooked in the pottery for the whole use of the pottery. And, well, why do we use lipid residue analysis? Well, the first thing we are using is very clear, since we're talking about all the time now today mentioned. We want to determine the original content of the pottery. We want to understand what is cooked in the pottery, what is processed in the pottery. And why do we want to understand this? Because we are curious, but we also want to understand the introduction of pottery into different cultures. Why this specific material culture came into different cultures, and what is the main reason, what is the motivation behind this. We can also understand the adaptation and the functional variation of the pottery. You can cook several different foods in several different forms of the pottery, or you can use one cup for everything to be cooked in the site. So we can separate with the form and the function of the pottery by analyzing everything that is in the pottery. These are kind of like the close-up questions that we can look into. If you want to get broad questions, we can also answer the discussion, like we can contribute to the discussion of ancient diets by answering what is in the pottery. We can also contribute to the culinary practices if certain a food was cooked in certain cups or it was cooked out of the cup, and what kind of food specifically used for the cup. So you can understand culinary practices in that way. And you can also contribute to the substances economy by seeing what kind of food was cooked inside and it was available in the close environment of this society. Or is it coming from somewhere else? Or is it seasonal? And one more step from that, you can also understand movement and connectivity of the past populations. If the food that you are finding in the pot is not present in the close environment of the society, where did it come from? Is it coming from the neighboring society? Are they in a relationship? Are they in a trade relationships? Are they in a connection? Is it actually affecting how they're living their lives, how they're using this material culture? So what can be identified through lipid residue analysis? We can answer a lot of questions about what are we actually finding in these pots. We can analyze several animal products in the pots that are non-ruminant, which are pig and wild boar, ruminant, deer, cattle, sheep, goat, and dairy fats. Dairy fats could be also butter, cheese, and milk. It's very difficult to separate. We can also analyze aquatic organisms, and we can differentiate marine resources from freshwater resources. And we can analyze biomass. We can analyze plant products to a certain extent. And that could be cereals, starchy plants, leafy vegetables, raisins, and tars. And for these identifications, actually, we use different kind of protocols to extraction. So not every analysis that you ask will give everything that is listed here. That's why we need a very specific research question from you. Well, after all this is great. Sorry. We are coming to the sample selection, which is very important, actually, for the research, because we can just select something to be analyzed. We're just going to get random answers, but what it means, it really depends on the archaeological context of the pottery. So I can give you 4 main criteria for this that you can apply if possible, and if not, then we can discuss more possibilities. One is that shards coming from secure contexts, and if possible, contexts that are already dated. If I know the pottery is coming from a kitchen context, if I know if the pottery is coming from a burial context, a household context, or a workshop, that will give me a lot of information on what I need to look for on this pottery. And I can identify the function related to archaeological context of the pottery. Otherwise, we will have, as I said, an information that is there, but if the archaeological context is not there, there's no in-depth storyline for this pottery. Second is that shards that are less processed, preferably the ones that have no glue, nail polish, or writing on them. This is to avoid contamination. And I know this is very difficult. We all been there. We wash the pottery, we wrote numbers on them, we store them in plastic. But if we can eliminate that, that would be a lot better, because I will talk about contamination later on again, but this will give you missed results, because I will pick up all the nail polish that is in there. I will pick up all the glue in there, all the writing or the touching that comes from your fingertips. So if we can avoid that, that would be very nice. And if you are at excavation and you know that you want to analyze certain kind of pottery, please use gloves and try not to touch them too much. You can, of course, wash them very gently. That is no problem for me. You can dry them. And if possible, if you really know that you want to analyze this pottery, wrap it in aluminum foil before putting in the plastic. Because I also can detect plastic in that. And sitting in the plastic over a long time is actually a big contamination problem for us. And the third one is that shards are big enough to be drilled without losing of their specific morphological and typological characteristics for future research. This is also not always possible. There were times that I had to crush the whole sample just to get enough material to be analyzed, but I can only do that in a secure context, that I know more material is there. And because it's a very destructive methodology, that I will drill into your pottery, I will make holes in your pottery. So you have to be sure that there is enough left for further research. And also you want to be sure that you photograph, you document it, you draw your pottery, you did everything else that needs to be done before lipid residue analysis, because after that I might just give you pieces of fragments that you can't just do anything with it. And fourth one is that shards represent the pottery variation of the site and or context they are coming from. This is again very difficult if you're dealing with small amounts of pottery, but if you are giving me a bunch of pottery that you think that represents an assemblage, that will give us a better understanding of the pottery use. And if it's only one or two pottery, I can still give you results, but we cannot talk about site function. We cannot talk about whole assemblage. And if you are doing that, please, please, please mention that in your report, that this is only for this pottery or for this context, that doesn't apply to the whole site or whole culture. So this is how it looks with the sampling. I am drilling into the pottery. On the first one, you can see the hole there. So I need to get, around 1 gram of pottery powder to be able to run all the analysis that is there. And also to make sure that if something goes wrong in the lab, that I have a backup material, that I don't have to go back to you, ask for more or destroy for more. And this is how it's stored in the lab. And this is the lab extractions. As you can see, this is from my PhD research, actually. This is the Swifter bound pottery, hunter-gatherer, towards agricultural pottery. The black part you see is actually the fat that is extracted from the pottery. So the preservation is amazing. And this is the machines, instruments we use: gas chromatography, mass spectrometry for molecular analysis. Gas chromatography, isotope ratio, mass spectrometry is for isotope analysis. And this is how it looks with the molecular analysis. This is I'm just going to pass this because if we don't have time. Okay, limitations. Like every methodology, we have limitations. Degradations is the first one that you were asking before. If the pottery is exposed to different kind of environmental events, environmental conditions, the lipids can change their chemical structure or they can disappear. So that is something we have to keep it in mind. And how we actually prevent that, we cannot prevent that. We can only do more experimental research in controlled environments and then compare to our data. The second one is contaminations. Contaminations can also happen from the soil that is buried to, or human sources. Mostly when we are handling the pottery, when we are excavating the pottery, as I said, the plastic you put in, or the material that you use to store with another material that you put next to it, or your sunscreen that you use on your hands, or if you do other stuff and then you don't wash your hands, we can all see that. So please be careful. And identifying sources with low lipid concentration is like one of the biggest ones, actually. And that actually wraps all the point of this workshop. When you're identifying different food resources, some of them has low lipid concentrations, like plants. If you're identifying more marine food resources, they are very high on lipid concentrations. And if they are cooked together, the marine food resource will mask the plant resource. So you will not be able to say, okay, there's fish, but there's also cereal in this pot. So for that, you need other analysis. You need interdisciplinary studies, which is, today we all heard about it. There are different ways of doing it. And this is why we're all working together to be able to understand one function of the pottery for you. This is it. Thank you.